Let me just, yeah, I'm recording, but I'm gonna switch screens. Where did they put that thing?
Did you have to pay for that domain name?
Which one? Palmer, a long time ago, yeah, palmerlab.org.
You have to continue paying for it?
Yeah, it's like, I don't know, it's $10 or $20 a year or something. It's kind of negligible, you know? But yeah, I just thought, like, if this is available, I'm taking it because Palmer is a really common last name, you know? And I didn't want to wait.
I think like several presidents ago, the old UBIC president still paying for the old UBIC website that we no longer use. Now we have a new one. I think
They're not very expensive. I think the first time I registered it, I got it for like 10 years or something. Okay, so I don't know if it's recording. It's recording, but I think it might be recording my notes. But anyway, they'll deal with it, right? Like this, you know, this version. If it records that, it'll be okay. All right. So all of this has been recorded. Do you want to introduce me or something, or?
Yeah, I do.
Okay, go for it. Yeah.
So the recording. Hello, everyone. Welcome to the first UBIC Talk Talk of Spring Quarter. Today we have Dr. Abraham Palmer presenting his talk, The Center for Genome-Wide Association Studies in Outbred Rats, as seen on the website www.ratgenes.org. Dr. Palmer is a professor of psychiatry at UC San Diego and vice chair of basic research. He obtained his PhD from UCSD in 1999 in biomedical sciences and is now a professor and researcher. Dr. Palmer's research focuses on developing a relationship between genotype and phenotype, especially behavioral phenotypes. His lab conducts genome-wide association studies to identify loci in DNA associated with behavior. Through utilizing strains of inbred and outbred mice, they study the effects of different gene recombinations on behavior. An ultimate goal is to investigate the behavioral phenotypes related to drug abuse, anxiety, and depression, allowing the applications of insights from the rat and mice research to human populations. In addition to research, Dr. Palmer is a 2020 Fellow of the American Academy for the Advancement of Science, 2020 Fellow of International Brain and Neurogenetic Society, and 2019 Fellow of American College of Neuropsychopharmacology. Please give a warm welcome to Dr. Palmer. Can you take a walk backwards? Oh.
Probably. Okay, great. So I think I'm going to sit because it seems a little nicer way to do it. So great introduction. Thank you for inviting me here. Let's see. Can I advance my slides? Maybe this will do it. Okay, great. So my lab, as you heard from that introduction, is interested in the relationship of genes and behavior, right? So we want to understand how it is that differences at the DNA level could give rise to differences in behavior in people, but we use animals as a model for a lot of that work. And we're interested in that because we think what's going on at the most basic level is differences in DNA give rise to differences either in the composition of individual proteins or the expression, the kind of the abundance of individual proteins. And that changes the way cells work, often, but not always neurons work. And then those neurons form constellations, brain circuits, and those brain circuits are the things that actually produce behavior. Okay, so we want to understand all of these levels. And the way that we're going to figure out about these intermediate levels is by identifying differences here that are correlated with differences here, and then using that information to hopefully fill in those intermediate steps, right? So that's saying a very broad focus. One of the things we're doing, one of the big things that we're doing is we have this Center for GWAS and Outbred Rats, okay? And that has a website, you've heard, ratgenes.org. And that's a center, so the way research like this gets funded is you write grants and you get grants, and the grants allow you to pay salaries of people so you can actually do something. And so back in 2014, I first got this grant to set up this center, and I'll describe kind of what that center has been doing for the last few years. And we've managed to get not just that grant, but a bunch of additional grants that kind of are all funding this project. So it's like a whole lot of different grants with lots of different people working on it, mostly in the US, but we actually have some satellite sites in like Ireland and Scotland and Italy, and we've got some German collaborators now. So kind of mostly US based though, and that's because funding tends to follow countries. So, and so the key elements of the center, and which I'll describe on the following slides, we can produce these rats, HS rats. I'll tell you about these rats. They're kind of special rats that we produce and distribute to different people. The people we distribute them to phenotype them, which is to say they measure things about those rats, okay? And then we here in San Diego genotype them. So we get tissue samples, typically from the spleen, but it could in principle be any piece of tissue from the animal. And we extract DNA from that. And then we can process that DNA in a very specific way that gets us genotypes, which is to say genetic differences. So what we want to do is identify sites at which not all the animals are the same, but instead where they're genetically variable. And it turns out there's millions of sites like that Most of them don't do anything. Some of them do something to other phenotypes. A very small fraction of them probably do something to our phenotypes that we're interested in. So we're going to have to analyze that data, which is the last step, analyze that data in a way to identify the true relationships between genotype and phenotype, keeping in mind that we're running lots of tests and there's lots of potential for false positives. So kind of if you had a stats class balancing false positive and false negative errors, to try to figure out actually where in the genome and maybe what genes and what specific parts of those genes influence these behavioral traits or really anything that we've measured. Okay, and so to do that, we have to have a lot of kind of statistical infrastructure. We have to have a big database where we store all of this phenotypic information, the genotypic information, et cetera. And then we have to produce kind of human readable reports about all of this. And so we've put a lot of effort into kind of setting up those steps. And here in San Diego, some of the rats actually are phenotyped by other labs, not by my lab. But the main thing that happens here in San Diego is all of this genotyping stuff, which has some wet lab elements to it, as well as some dry lab elements to it. And then all of the database and analysis stuff all happens in my lab here in San Diego. So this is sort of the nerve center of this big project. So let me tell you a little bit about these rats, because the rats, as I said, are a little bit special. Back in 1984, actually, sorry about that. Back in 1984, somebody in Washington, D.C. area decided that they wanted to have a genetically heterogeneous population of rats. They wanted to have a population of rats where not all of the rats were genetically identical to one another. Oftentimes in animal research, what people do is they actually inbreed for lots of generations to make animals that are genetically identical. So you probably were told that you shouldn't inbreed, like you shouldn't mate with close relatives, and that's true because you'll have lots of unhealthy offspring. But if you don't care about that and you keep inbreeding and only select the healthy individuals from each generation, you can actually produce an increasingly inbred population. And after 10 or 20 generations, mathematically, you should lose all genetic variation, okay? Because every time you're sampling, if you have a small number of breeders, you lose genetic variation with each generation. And eventually you end up with individuals that are homozygous at all loci, so they have no genetic variability left. Of course, there's always new mutations every generation, so that creates a little bit of a problem. There's some background variation, but essentially all the animals are genetically identical. And that's super common. That's been happening for 100 years with mice, with rats, with flies, with various plant species, et cetera. People love these inbred strains. So in order to actually fight against that, in 1984, these people decided we're going to take a bunch of inbred strains, 8 of them shown up here with different colors, and we're going to deliberately mix them, so make them not inbred anymore. And you can imagine you set up breedings, and eventually you get a population like this, where you have a whole bunch of individuals and each one has various genetic information for these 8 founders, but none of them are inbred anymore. And they're all genetically unique. Each animal will be genetically different from every other animal. So that's like a normal population. That's like a normal human or any kind of natural population where there's genetic variability. And to do any kind of study where you're going to relate genotype to phenotype, you obviously need genetic variability. If everybody's the same genotype, there's not much to say about the phenotype. Anything that's different about them phenotypically isn't because of the genotype, because they're inbred, right? So we need to produce this outbred population. So this was done in 1984, and they've been passed around. I think I have a slide for this. They've been passed around a bunch. So in 2003, they were sent to somebody I know named Ava Reedy at Northwestern. And then in 2006, the current collaborator and maintainer of these things, Leah Solberg Woods, got them. And she had them for a while in Milwaukee, and then she moved to someplace in North Carolina, I think. And since this center got set up in 2014, we've tried to maintain lots and lots of breeders. So initially we had, I think, like 46 breeder pairs. Currently we're operating with 92, or I think it's even 120 breeder pairs now. So we have lots and lots of breeder pairs. Obviously you would fall back into inbreeding if you only have one or a few breeder pairs, right? So you need to have a lot of breeding pairs so that there isn't much genetic drift, so that unique loci aren't being lost, okay? And so we have a lot of breeding pairs, which means we can produce a lot of rats. And as you'll hear in the subsequent slides, we then ship a lot of those rats to people who are going to phenotype them and send the DNA to us. And then, of course, they maintain some of the animals that are offspring to breed the next generation. And remember that, like dogs and cats, rats have litters of like 8 to 10 pups per litter. So you get a whole bunch of animals from a breeder pair. Okay. And you don't care, but it turns out that during the pandemic, we got worried about what would happen if this colony got lost. So we had a lot of embryos collected from lots of breeder pairs and actually frozen, and they're now sitting in liquid nitrogen in 2 different states, so that even in the event of like a limited nuclear war or something, we'd be able to reconstitute this population. I don't really expect a limited nuclear war, but you never know. Okay, so here we have this animation, which is kind of cool. This guy is Dan, who's a postdoc in the lab. And if I can get it to play. Like this. Yeah, there we go. So he just wanted to simulate what would happen to an individual chromosome that exists in a lot of individuals over time. So as you're breeding these animals over time, what happens is these 8 different founder chromosomes get mixed with each other, okay? And we could sort of track that because we could actually measure genetic differences in individuals and we could watch that mixing go on. But more importantly, we wanted to know if what we were observing in modern times was sort of consistent with the amount of mixing and the kind of mixing that we think would have happened. So he set up simulations of how those chromosomes would be expected to mix over a lot of generations. And then he ran those simulations and compared them to our real data. And we were reassured to find that, yeah, the amount of mixing looked consistent with what we were observing. So probably this whole historical record back from 1984 to the present time was mostly accurate. You never know if it's completely accurate, but it's mostly accurate. What we think we have is really what we have. And as you'd expect, after a bunch of generations, those big chunks of chromosomes that came from the inbred founders, remember with each generation, chromosomes recombine when you make gametes, and so they're getting mixed up into smaller and smaller blocks. So now if we look at a modern day individual and we look across the chromosome and we try to reconstruct for each segment, which founder did it come from, we find lots of transitions where very frequently you transition, okay, this looks like it's from founder 1, but now as we walk along that chromosome, this segment looks like it's from founder 4, and et cetera. And that's just what we'd want, because we want, remember, to look at genetic differences and to associate a genetic difference with a phenotypic difference. And so if we see a genetic difference associated with a phenotypic difference, we want that to define a narrow area where that region seems to be critical for the phenotypic difference. And by producing all of this mixing, that area becomes narrow, because if we went very far in either direction, the markers that we measured would be from other founders. Okay, so it means that this little piece that comes from a particular founder seems to be critical. So all those generations of breeding, which obviously we didn't start, that started a long time ago, but those really benefit us because it allows us to localize these signals to very small intervals. Okay, and I'll talk a little bit more about that and show you some pictures if that's not completely gelling. So you get to a certain point in life and you start to actually realize that org charts have a place. Okay, so this is like an org chart of this center, just to give you an idea of sort of the interactions between different parts of the center. And the key thing to take away from this is here's this breeding core. This is this woman I mentioned, Leah Solberg Woods, who actually produces these rats. So she's producing thousands of rats a year. She's almost like a little academic company that sells rats. And she sends these rats to different groups that do phenotyping. So these are groups that have a very different expertise. They know a lot about measuring behavior in rats. And I'm going to tell you a little bit about what they measure in subsequent slides, but they measure things about these rats. And they're consuming anywhere from like maybe 200 to 800 rats a year. Sort of, they're getting these rats. They do this very detailed phenotyping on them. They send us the phenotypic data and they send us the DNA. And that comes here to us in San Diego 4C. And then we're going to analyze that tissue sample to get genotypes, to get genetic differences. And we're going to analyze the kind of raw behavioral data that they send us to try to figure out what are the key phenotypes or the key behavioral differences. Yeah.
Out of curiosity, what happened to the rats after their phenotype?
Well, eventually we kill them, right? And actually, as I'm going to tell you in a few slides, once we kill them, we cut them up and we take lots of tissues, because there are all kinds of people in the world that want to study things about rat tissues. And so it's sort of like the idea of using every part of the buffalo, that once we're done with these animals and we've got all the behavior we want from them, then we collect all this phenotypic information from them. So, and we kill them very quickly. And if you're concerned about the welfare of the animals, let me point out that UCSD has rat traps all over the campus, and the animals eat like warfarin, it's an anticoagulant, and then they die from internal hemorrhaging, and it takes a day or two. Something we would never be allowed to do in the lab, but there's all kinds of animal cruelty going on. There's also people eating meat, et cetera. So I think what we're doing with these rats is pretty humane compared to what goes on with animals all over the place. All right, so a bit about the phenotypes that we measure. So I mentioned that there are these 3 key groups that do phenotyping. I know you guys don't really care about rat behavior particularly. I'm going to come over here so I can read my slides better. But just to give you a sense, some of them go to the University of Buffalo, which is in Buffalo, New York. And this guy looks at intermittent cocaine self-administration. So remember, maybe I didn't even say, most of the money comes to study drug abuse. We want to understand sort of the genetics risk for drug abuse. So he has these animals self-administering cocaine, and this is kind of just what you've seen like on a Nova special or something. The animals are hooked up, they have a catheter going in that goes right into a vein. They can press a lever, they get cocaine. Cocaine feels really good, so the animals learn to press that lever a whole lot. And then you can see like how badly do they want the cocaine by saying instead of 1 press, now you need to give me 2, now you need to give me 4, now you need to give me 6, 8. And you can see at what point is it not worth that infusion of cocaine, right? And so early on, the break point at which they no longer start taking cocaine is kind of low, like maybe after 20 presses, they're like, yeah, whatever, who cares about cocaine really? But then after they've had a long history of weeks and months of taking cocaine, they'll hit that lever like 100 times to get a single infusion. So sort of the value of cocaine or the amount of work they're willing to do goes up a lot. What's interesting about this is that in these HS rats, some of them actually don't really care about cocaine. Some of them will take a little bit of cocaine on the first few days, maybe on subsequent days, they'll take it once or twice, but they really don't care. And this is like free cocaine, they don't care, okay? Others of them though, as you'd expect, escalate their cocaine use. So they keep using more every day, and they eventually get to a point where they're using huge amounts of cocaine. So we're interested in why that is. And in fact, in humans, we know that's true. Most people that take cocaine never become addicted to it. They use some, they use it, they could probably get more, but they just after a while lose interest in it and move on to something else. But a few people have this big problem. Maybe about 20% of people that use cocaine get into a lot of trouble because they escalate their use and they kind of get into a stage of uncontrolled use, do all kinds of terrible things to try to get more cocaine. So we're interested in genetically, what is it about the brain? We think it's probably got to do with the way the brain processes reward. What is it about the brain of these animals where some of them aren't very interested in cocaine and some lose control? Yeah.
How do you legally get cocaine to get to that?
Oh, we have a license. We have a DEA license, so we can like order cocaine from Sigma Chemical Corporation, and they send us cocaine. Now, keep in mind that rats are small, and so they don't actually use that much cocaine. So the amount of cocaine we buy, I don't want to profess to have a great knowledge of street value, but maybe we have like $500 of cocaine or something like a street value. Like it's not in the scheme of things that much. So yeah, just like a pharmacy or a doctor could legally get various scheduled drugs, we have license and we can buy these drugs. And as far as I know, like there's lots of labs that have that kind of a license. As far as I know, there's been very little problem over decades of people stealing cocaine that they buy legally through these programs, because it's just not that much. Okay, so that gives you a sense for what Paul is up to. There's this guy, Hao Chen, he does pretty similar stuff, except it's with nicotine. So again, these animals are able to consume nicotine. In this case, they actually get it from their drinking water. Oh no, I'm sorry, they don't. No, they get it IV also. And we can't give it to, or we don't give it to them as like a vapor or a smoke, because that's not a very intuitive way for an animal to take a drug. But they can definitely learn to press a lever. And some of them take very little nicotine. They don't like it. It seems like it makes them sick, maybe. Others take more and more nicotine, and after a while are willing to work really hard to get more nicotine. And then the third one is a little more complicated, but we're interested in kind of how animals, it's a little bit about behavioral regulation. So for example, we look at whether an animal wants a small reward that they can get right now, or whether they're willing to wait in order to get a larger reward that's delayed. Okay, and we can actually kind of figure out what is their function for valuing current versus future rewards, because we can change the delay between the current and the future reward, and we can also change the disparity. So like we could make the future reward really huge, or we could get just modestly larger than the current reward. And it turns out that animals, including humans, but also rats, primates, pigeons, et cetera, they have this kind of very characteristic hyperbolic discounting function, where the longer you have to wait for something, the less valuable it is. And so what we want to do is find out the constant of that hyperbolic function, and we can assign a constant to each animal. And then it turns out that's quite different among the animals. So some of them really don't care about future rewards, other than are very interested in future rewards, and they're willing to wait. And again, we want to know why. And there's a lot of evidence that in humans, that kind of a behavior is related to the risk for substance abuse. And so we're particularly interested in that. But it's also probably interesting, it's probably relevant to lots of other interesting things that people do. Yeah.
When you say like a bigger reward, say
Yeah.
you gave twice the amount of sucrose.
Yeah.
That's not, is that really like equivalent to say twice as nice of a reward? Because like the value kind of diminishes.
I mean, we think so. They all will wait if the reward is big enough. They'll wait at least sometimes for these bigger rewards. So yeah, exactly how do they process that? And by the way, it's just water. The animals are mildly water deprived. So they haven't had water for like 8 hours when they go into the session. So they're kind of thirsty, and so they're working to get this water.
Yeah,
I mean,
I was just wondering how you
yeah,
quantify
it seems to work. In humans, you actually ask people questions, often about money. Do you want $5 now, or do you want $8 next month? Right? And you could imagine you would change the the delay and the disparity. So anyway, all I really want to communicate is we have these behaviors that we're interested in, and the measurement of the behaviors is pretty complicated, and oftentimes involves weeks or even months of behavioral sessions that the animals are in. So we get a lot of data, and you can imagine you can extract like one quantity from that, like this hyperbolic discounting function, but there's a lot of data there. And one interesting thing you could do is think about different ways of extracting data from that, different ways of kind of defining phenotypes to analyze them differently. Or if you found that they were different for that hyperbolic function, you might ask, well, what is it particularly about the reward disparity? Like, is this more relevant to the delay, or is it more relevant to the difference or something? There's a lot you can do with that data. And we have all of that data actually stored in our database. And so there's potentially a lot of projects to be done there. The fact that we set this up meant that it was really easy for us to establish new collaborations with other groups where we'd say, hey, we can send you these rats, we can like genotype and analyze these rats. So if there's something you think is interesting to measure, and if you can convince the government to pay you to do that, we can easily kind of support your study. And that's led to lots of labs that are collaborating with us that also have funding to do stuff. And so a lot of these are about substance abuse, which are kind of the ones that have these red UO1s in them. But there's other things, like we're working with a group that's interested in glaucoma. We have some stuff here with alcohol. We have some stuff with bone matrix, whatever that is, something about like bone quality. So lots and lots of people have kind of joined this center because we have this infrastructure set up to do this rat production and genotyping and phenotyping and analysis. And that's been good for us because it means that we get really big sample sizes. And so now here's kind of an updated version of this thing with a lot of new projects that have been added to this, looking at other phenotypes. Again, a lot of them are drug abuse, not all of them are drug abuse. And then we're doing some stuff that I've shown over here kind of on the side because they don't exactly fit into this idea of like sending people rats and getting data. But for instance, we're looking at other kinds of genetic differences in these animals. So typically people have looked at single base changes in the animals as the thing that causes differences. But there are also, it turns out in the genome, little repeats, like sometimes a 2-mer, a 3-mer, a 4-mer, like a series of nucleotides that repeat. And those are quite mutagenic. And so oftentimes over short periods of time, animals will go from, say, having 2 copies to 3 copies to 4 copies. Some of those change things about behavior and other phenotypes. So we're now measuring those, which requires a different sort of a technique. There are also regions of the genome that get duplicated or deleted. And so some people might have 1 copy of a particular region that might have a gene or several genes in it, but other people may have 2 or 3 or 4 copies of that. And that also can change the behavior of the animal. So we're using other approaches to measure that. We're also getting into this cool idea that you can look at gene expression in individual cells. So there are increasingly sophisticated technologies that are used to separate the cells, or sometimes just the nucleus of the cells, into individual micro droplets. And then each droplet has unique DNAs that you use to barcode everything that comes from that droplet. And then when you sequence it, you can figure out, oh, all of the reads that I'm looking at here came from this droplet that has this barcode in the sequence that I added to the sequence. And that allows you to get data sets where you have gene expression from thousands, conceivably millions of different cells, and you can figure out exactly which cell it came from. So you could start to identify maybe there's a difference, and it's not just in the expression of genes in this animal, but it's in the expression of genes in a particular cell type, and maybe that's more interpretable and actionable. Yeah.
What types of cells do you look at? Is it primarily neurons or is it other?
Yeah, we would mostly look in brain, and so the brain is like very round numbers, kind of half neurons and half glial cells, which were traditionally thought of as just sort of like boring support cells, but it turns out that they have a lot of signaling functions also, kind of slower time scales. But so we would get a chunk of the thing you can't do easily is select which cells you're going to look at. You get a chunk of tissue, and then you're going to look at whatever cells are in that chunk of tissue. So anyway, so that produces some pretty rich data sets, and you could imagine there's a lot of bioinformatics to be done with that kind of data. And so what I wanted to tell you is that we now have done this with about 10,000 rats, and we have funding, I think, right now for about 6,000 more, but we've got more grants coming in all the time. So this is really a big, big data set. This is like the biggest data set of its kind for either rats or mice that exists in the world anywhere. And so there's a lot you can do when you start to have that big data set, but you also realize that you've got a big data problem, and all of the things that happen with big data, like the quality control elements and the storage and the processing and, the analysis and et cetera. So it brings you to talk to people like yourselves. We have a lot of stuff to do. I'm going to kind of go through these quickly. I was going to just show you a little bit more about exactly what data we collect from animals. So in this example, this is a group that looks at heroin IV self-administration. So really similar to what I told you about before. But I don't want to bring you through all of this except to say that you see there's about 48 days of measurements. So there's a lot of data. Each session, I think, is maybe 6 hours or something. So we have data at a millisecond resolution of what the animal was doing in these 6-hour sessions over 48 days. It's like a lot of data, right? You can imagine. And not only do we measure how much heroin they take, that's an obvious thing to be interested in, but we're measuring other stuff like pain sensitivity and how irritable they are and different kinds of things, how they're behaving when they're in withdrawal. And then at the end, we actually stop giving them heroin for a while and then look at how much they want heroin when they come back to it a week or two later. So after all of the acute withdrawal stuff has happened, like the immediate, kind of feeling like you have the flu sickness is over. But what we know about humans that abuse drugs is that even months later, they still have this incredible risk of relapsing to drug taking. So it's not that they're feeling, in this case, dope sick or something, but something about the brain has changed, that there's this constant elevated demand for the drug. So we're interested in looking at that also. Blah, blah, blah. Okay. A lot of detail here that we don't need to look at. Wow, okay, I could have spent a long time on that slide. This is just a quick one to show you what I said before. So this is now animals taking oxycodone, which is an opiate drug similar to heroin kind of. And you can see that we have some animals, so this is over days, how much heroin do they take, basically a measure of a lot of things, but essentially how much are they taking, how much oxycodone, sorry, do they take? And you can see some animals increase their use over days and other animals don't. And so here's like a plot looking at that, where you see there are some animals that really don't seem to increase their use, like they're not very vulnerable. And there's some animals that seem quite vulnerable. And so we and a lot of other people are really interested in genetically, what is it that makes these animals different? Skip that, that's more about the single cell. I'm going to skip this, I told you about this a little bit. This is about detecting these different kinds of variants, like these tandem repeats and structural variants. But I guess what I want to communicate is we have lots of collaborators. So here, Graham is over at the Salk and Francesca's in medicine. Melissa, I think she's got some kind of appointment over here. I don't know if you guys have met Melissa or not.
Yeah, she was
Yeah, exactly right. Okay, so you guys know Melissa. We're talking with Melissa. Jonathan is also in psychiatry. So just a lot of people that we've drawn into this because there's a lot of different kinds of expertise you need to do this work. Here's a little bit about our database. So we have a team of like 4 full-time bioinformatics people. So they sort of have bachelor's or master's in something like computer science or bioinformatics. And in fact, we're always hiring more in case any of you are seniors or.
I'm a senior. I had a job.
Yeah, all right. So yeah, so we're always advertising for more of these people. But we want to keep this data in a very organized way. We're increasingly being approached by people that do machine learning stuff. And so they want to like gobble up all of our data and try various machine learning approaches with it. So we want to keep it in a very documented way. There's a lot of quality control issues. You can imagine that all that phenotypic data we're getting from these labs, there are all the time like errors or mistakes or out of range values or all kinds of problems. So when we take it in, we want to be very careful about the data that we're bringing into our database and have a lot of rules so that we don't take in crappy data. So anyway, so that exists and that's of interest to you guys, I would think. Yeah, okay.
So say you encounter some stuff that it has some fishy outliers.
Yeah.
Do you just prune the outliers or do you just like prune the whole thing?
We'd like to figure out why they're outliers. So often if we go back to the lab, they'll say, oh yeah, there was a blackout that day, or they were jackhammering right outside of the building that day. So that, maybe that explains why they were outliers. But there sort of could be like the measurements are true, but they're weird and maybe we don't want to use them. Or the measurements are not true, like there was something wrong with the equipment, or with our pipeline for importing the data. And maybe that's even correctable, but you would think about it differently. We don't want to throw out values that we think are true values for which we have no reason why they were outliers, but they don't look right. But if they're, 10 standard deviations out, we're not going to use them, right? We're going to assume something's got to be wrong with this because you really shouldn't have values that are 10 standard deviations out. So it's a bit of art rather than science. Okay, so this is gross. I'm sorry to show you something gross, but here's a dead rat cut open, and this is 2 kidneys. That's what you're supposed to have. You're supposed to have 2 kidneys. Do you see what they're called? Kidney beans. Like a rat kidney looks almost exactly like a kidney bean. Okay. And this is a rat that has only 1 kidney. And it turns out that about 2% of these animals only have 1 kidney. It's like a congenital defect where they don't form their second kidney. And so we can then look at the relationship between genotype and whether you have 1 or 2 kidney. So this is sort of the bread and butter of what we're doing. Is here we have a bunch of chromosomes. All the chromosomes of the rat genome, they have 20 instead of 23. And here we have a sense for how statistically significant is the association between any one of these dots and the probability of having 1 or 2 kidneys. So each dot is a genetic polymorphism or a genetic spot at which not all the animals are identical. Okay, so it's something that we can measure that distinguishes some animals from other animals. And you can see that most of these have relatively low, and this is the minus log of the p, so like higher is more significant. Okay, it's just a convenient way to make a plot so that higher is more significant. Most of these animals, or most of these markers rather, are not really strongly associated with the 1 or 2 kidney thing. Okay, so these are sort of levels of significance we might expect to see by chance, at least in a small number of the tests. But very obviously, there's a huge peak here, right, where these things are very unlikely to be associated at that level by chance alone. So we actually are quite confident that that's the thing that's governing whether you have 1 or 2 kidney. And what we'd like to do is to drill down and to figure out as precisely as we can exactly what in that region differs between these animals that have 1 kidney and the ones that have 2. And this is a little bit complicated because we're pretty certain that you can have the risk allele for this and still end up with 2 kidneys. So it would be an easier problem if every time you had the thing that confers risk for 1 kidney, you had 1 kidney. That would be, we already be done with the project. But it seems to us that even if you have the most extreme risk allele, you only have about an 8% chance of having 1 kidney. And so that's made it a little bit harder. We need a little bit more statistics to unravel that, but we're working on it. Yeah.
And N of 110 for the affected, that's
Yes.
pretty small.
Exactly. So like I said, it's only about 2% of the animals that have 1 kidney, right? So we have few cases relative to controls. And so some of the test statistics actually don't behave well when you have a real imbalance between cases and controls. So we've used a particular package that's designed to be robust against that kind of a problem. And so it uses a different statistical test, like I can't remember what it is, but it's not a chi-square. Yeah.
Regarding the specific genes on like chromosome 14,
Mm-hmm.
have you guys tried like looking at, oh, maybe it's like one of those things where you not just need this specific variant, but also you need another one too. So you
Yeah.
guys look at the combinatorics.
Yeah, so that was one thought that we had, is maybe the reason only 8% of the ones with the risk allele actually have 1 kidney is because there's some second factor. And we've used various statistical approaches to try to see if there's a second factor like those. It's tempting to see something like that and think, oh, maybe that's like the modifier or something. I can't rule out that there's lots of little modifiers, but there doesn't seem to be any one principal modifier. We would have had power to detect that. And so far we haven't. We think it's probably stochastic, unfortunately. We think there's probably some non-genetic sort of random factor that actually decides whether or not you're able to form that second kidney, which is bad news for us because we're not good at measuring stochastic, right? But that's what it is. And anyway, I think we're going to figure it out, but it's a tough problem. What's more common is, so that's like a rare example where the phenotype is all because of one thing, it seems like. What's more common is lots of little things. And so if you look at things that maybe you're familiar with, like human height or human intelligence, which turn out to be very heritable traits, they're not due to 1 or 5 or even 10 different things. They're due to like thousands of things that make really tiny individual contributions. But that's why if you've ever had a friend who had tall parents, the friend was probably tall. If you had a friend with short parents, the kid was probably short, right? Because they inherit lots and lots of hundreds, probably thousands of things, each of which have a little infinitesimal effect on their phenotype. And so that's the case in rats. We, of course, have weighed all of these animals. So we have lots of data. At this point, we had 3,000. We now have many more thousands of animals where we know how much they weighed. And we can ask, what are the genotypes that influence weight? and you get lots of different things. So here we actually looked at weight and some other kinds of things like fat pad weight and some other sorts of things. But what we find is lots of different sites are implicated. This is sort of a threshold for significance. You can see lots of things are significant there. That's a cool data set, and we're actually doing various things to see whether or not the things that influence rat weight and rat BMI also influence human weight and human BMI. And so like we're working with Trey Eidecker to use networks to see whether or not we can relate those two things to each other. And we're working with some person at University of Chicago to use a different sort of approach to see if those things are related. This is what so here you see lots of peaks. This is what one of those peaks looks up when we zoom in. So this is only chromosome 1, only a tiny portion of chromosome 1. And when we zoom in, you can see we have a peak. Here's like this most significant signal. And we have some genes that are in that region. We have various ways of figuring out which of those genes we think is most likely to be causal. In this case, interestingly, there's a very famous gene, TCF7L2, which has a big effect on human BMI. And so we think it's very plausible that TCF7L2 might also be responsible in the rats.
How did you get named?
Oh God, it's a chaotic process. Most genes have been named by now, but TCF7L2, I wonder, transcription something factor 7, and then it was part of a big family, so it got a 7L2, I think. Not in a very organized fashion would be the easiest way to answer your question.
Like my genetics class last quarter,
Yeah.
every time she brought up a name of a gene, like, well, it's a weird name. I didn't mention that.
Yeah, you can see they all have weird names. They recently actually had to rename some genes because when you import a gene name into Excel, sometimes Excel will think like that's a date or some other thing and it'll change it. And that was such a problem and they could not convince Microsoft to do anything. So they literally renamed genes that their name is no longer misprocessed by Excel.
I've been through this too.
Yeah.
It's super sad that that would even be an issue, but anyway. And there's ways to defeat it, but the problem is people don't. Like there's ways of turning off that function of Microsoft.
Yeah, circadian rhythm's nice because there's like the clock gene and the time gene. So
Yeah.
that's really nice.
Yeah, sometimes people have named them in ways that are really, but typically names like this mean they didn't know what it did when they named it. So anyway, okay. And we're also looking at another kind of a trait, which is gene expression. So you could imagine that we can measure thousands of genes to see what level at which each gene is expressed. And then we can ask, what is the genetic variants that are associated with differential expression of that gene? And typically those are things right near the gene. So like body weight or single kidney, that doesn't have like a location in the genome, right? But the gene actually lives on a particular chromosome at a particular address. And so usually it's things that are very near that gene that influence its expression. And that's what the strong diagonal is meant to reflect. But not always, and many genes actually don't have heritable variants that influence their expression, but some do. And it's useful for us to know which is which, because when we're looking at a region like this, we would like to know, are there any genes here whose expression is heritable, where differences in their expression is heritable, because maybe that's what's causing the difference. So that's sort of a data intensive thing. So I have some projects here, but I don't think this even scratches the surface. And really, if this is broadly interesting to you, just like email me or talk to me in person right now and we can figure out sort of what you might want to work on and what your time horizon is, and et cetera. But just to give you an example, like I said, we have about 10,000 rats that we phenotyped and genotyped now. We actually use two slightly different approaches to genotype them. And we have yet to come up with a unified way of calling all those genotypes. So we think the two sets are pretty comparable, but we'd like to run them all through the same pipelines, that we have a set of genotypes where we're very confident that the first set and the second set are compatible with one another. So we need to kind of play around with some parameters to run some code. You know, some of these are pretty big jobs when you have 10,000 individuals to run some code. We have some truth sets where we're pretty sure we know the real genotype, and so we can use that. And we've run some of those animals through both the old and the new. So we can sort of use that to figure out whether things are working or not. But you can imagine there's some work to be done with that. And it's also important that it work. We have, I should look at my own screen. The genotyping pipeline is pretty slow. So even using kind of supercomputer like resources and using lots of processors and et cetera, it can take us days to weeks to call these genotypes. And so we really think there are some ways to improve the efficiency, like just allocating the right resources and the right amount of wall time, breaking the jobs up and parallelizing the jobs and et cetera. We have some people working on that stuff, but there's a lot more to be done there. We also want to make these things portable so that sometimes we could like send them to Amazon or something like that, you know, and not be dependent on the particular system that we're using. And so people are using Docker and other kinds of things to kind of make these portable. So we're working on that sort of stuff. When we import the phenotypic data, we were talking about this, there are lots of problems. So we already have kind of rudimentary QC reports that we produce. Every time we run a batch job and we import a bunch of data, we want to flag outliers and other kinds of suspicious values. But that could be improved. And since we have a bunch of different collaborators and each of them gives us really kind of qualitatively different data sets, we need to have a way of coming up with these QC reports, like working on the pipelines to avoid problems that are our fault, and then working on these QC reports to identify problems that are usually their fault, so that we can say, hey, this is screwed up, can you fix this for us or whatever. When we run an analysis, and I showed you some pictures of what those analyses looked like, right, with lots of dots and p-values on the y-axis, we actually produce a big report, and this is something we've been developing for years. And so it's an HTML report that's got lots and lots of different kinds of data readouts that we give back to our collaborators. But we have all kinds of features we'd like to add to that and improvements we can make to that. And kind of like it's increasingly interactive where you can like resort things and zoom in and out on things. So there are a lot of sort of like individual jobs, some of which are really probably pretty simple. Like they would be, I think I wrote quick wins here because they might only take a week or something to set up. And others would be really sort of like deep dives into improving these reports. But these reports we distribute to like, as you saw, more than a dozen labs. And we want them to be really good and useful. Our goal is basically that we're writing papers for people. Like we're giving them all of the figures and tables they need to write a paper, because the old approach was an analyst would like take requests and do things and send it back to them. And it was inefficient and repetitive for us. And so we're trying to just do all of that in an automated way and give people what they need. And that's cool. And actually, I think other people are starting to use these reports that we've made. So it's sort of like a pipeline that maybe is broadly interesting to people. This looks like the same slide. And I've got some others. I'm going to stop in the interest of time. Some of these others are a little more esoteric. I'll give you the slide deck and you guys can look at it if you want. But there's a lot to be done, basically. You know, like the key thing about our lab is we have some wet lab steps, but we have all of the dry lab steps. And so at every level, there's big amounts of really interesting data that we've got that we want to process and use as efficiently as possible. We want to make it accessible to other people to use in other kinds of ways, some of which we might not even anticipate. And so we have, like I say, right now there's 2, soon to be 4 graduate students. There's 2 or 3 postdocs. There's about 5 staff positions that are associated with processing the data. And then there are several sort of other faculty members that are part of this group. And so it's a big operation and we have big meetings. And we would love to have more undergraduates working on the data side of it. We have a lot of undergrads that are sort of working on wet lab stuff and data collection, but we haven't gotten as much interest, I think, because people just don't know how to find us in people that are coming from more like a dry lab orientation. So this is sort of some of the outreach that we're trying to do to get more people like that involved. All right, I'm going to stop there.
Could you go back like a bunch of slides
Yeah.
to the one where it was, I believe, in regards to like to opioid addiction, or like the resistant one? There's like
Yeah. This, yeah, I know the one you mean. This one, right? I had it for a second. That one. Yeah, right.
How did you? So when you say resistant, how did you group them together?
Yeah. So this is this actually variable and this variable are not the same, but so this is a z-score that z-scores together several different aspects of their behavior that we think are sort of on a continuum from more or less addiction prone. Okay. And so you can see then that we just can easily put a line between them because it's pretty bimodal. Sometimes it's not fully bimodal and you kind of arbitrarily decide how to break this group into that group. Okay. But anyway, you can just put a line and say, I'm going to call this group and plot them separately from that group. That's not a statistical procedure, but it's just a way of sort of illustrating what how the data are behaving. And then this here is just a particular kind of behavior escalation on particular days of testing. And you can see that this also shows nice bimodality. Is that kind of what you're asking?
Yeah.
Yeah.
Is there any like stuff? So since now we know like the genetic variants,
yeah,
like plenty of GWAS movements. Is there any like stuff to do? Any like current stuff where you essentially prescribe in clinical trials opioids or painkillers to people regularly versus, and then you look at their responses.
Yeah,
And
it's
then you kind of maybe,
there definitely are people studying substance abuse in humans, obviously, right?
yeah.
Some of those things are harder to study. So for instance, cocaine, which is actually what we're looking at here. I hope I didn't say something else, but it's pretty hard to get cocaine abuse subjects because they don't tend to interact with health systems. And in fact, the people at highest risk for cocaine tend not even to have medical insurance, right? And so it's harder to get that data. People also don't typically like volunteer to say I'm a cocaine abuser because they know that they're committing a crime and so they don't want to like identify themselves. So it's been hard to get big, powerful data sets of humans doing that. For opiates, where sometimes a doctor is writing it, it also can be hard because what happens is the person starts out getting a lot of opiates from the physician, but then increasingly they start buying them illegally because they can't get as much as they want from the physician. And they're obviously not going to tell the person who might write them this, drug that's become very valuable. They're not going to say, oh, by the way, I have a problem and I'm abusing these drugs you're prescribing me. And so again, it can be hard to get that kind of information.
Yeah, I meant like using that information kind of reinforce treatment.
Yeah, I mean, there are many things that are being done. I don't mean to say that there aren't many smart things being done in human genetics. There's also a lot that goes on in humans. You're not sure what's really going on. You can't get people to join the study when you want them to. There might be a lot of environmental or other things that are exacerbating the risk, but that are not genetic. So there are a lot of problems with doing these sorts of studies in humans, and that's why we sort of like doing them in rats, because it's easier for us to have consistent conditions, and therefore we're really studying the biological basis of it without looking at sort of the sociological or epidemiological issues. Yeah.
Do humans also have the one kidney phenotype?
Apparently a few humans do, but it's much less common. So there are people going around that only have one kidney. Many of them don't even know about it until somebody does a scan, because it turns out you only need one, which is why people can sometimes donate a kidney and they'll be fine. Yeah.
Couple things.
Okay.
If you would like to, so first thing, are you okay with us posting this?
Yeah, definitely. Yeah. Great.
Second is, if you'd like, you can, if you're okay with this,
Mm-hmm.
I'm sure that people would also like to look at the slides. so you can-
Yes. I was going to give you the slides. Yeah.
Okay, great. And then the
Yeah.
third thing, when you send me the slides, if you'd like, you can also do like a write-up
Uh-huh.
or just advertising like what people you're looking for. And
Yeah,
just
yeah.
general advertising.
Yeah, sure. And if you have some examples, have other people done that?
I can
Yeah,
you
send me
that.
some examples and I'll kind of, examples of people who did it well. And I'll try to imitate the examples of people who did it well.
I can do that.
Yeah, perfect. Okay, good.
Okay. Becky, so more.
No, just the other thing is the people here, come to DVM tomorrow.
Oh yeah.
I think it's scheduled during my last class.
6:30.